Hello. Lecture number 2, we're going to talk about how the cell works. The cell is made up of tons of different substances, molecules that make up the cell membrane, small proteins inside that form basically small organs inside the cell. The cell is it's a single-celled animal. Each one of our cells thinks that it is an amoeba or something, a single-celled animal floating in an endless sea. It turns out that the endless sea is the fluid that our body produces from the blood flow to wash over the cell and keep it moist, give it oxygen, give it nutrients, take away its waste products, and so it just happily sits there and does its job for us because we take care of it as well. So here we're going to go. Number of different kinds of cells. First of all, some terminology here. Site means cell. So since osteo is bone, an osteocyte is a bone cell. A skeletal muscle cell is a myocyte, because myo means muscle. So cyte is the cell. A blast is an early form of a cell that creates things. You have something blast. Osteoblasts create bone. An osteoclast removes tissue. So if you have a bump on your bone because it was broken and healed, the osteoclast will eat away the bump while the osteoblasts are healing the break, and the remodeling of that bone is possible. Here we have erythro, means red cells or red blood cells, erythrocytes. Fibroblasts, these are cells that make fibrous connective tissues inside your body to hold you together. Epithelial cells, these cuboidal cells here that we're going to learn about later are a lot of them are in glands. They secrete stuff. There's a skeletal muscle cell, long and skinny. Smooth muscle cells shaped a little bit differently. Lots of different kinds of cells. Here we have a fat cell, which is mostly filled with fat, just clear fat with a little nucleus over here off to the side. Here we have immune cells, cells that fight disease. A macrophage, we'll see this phage or phagocyte, that term, it means to eat. A phagocyte or macrophage will actually surround and engulf a bacterium or a dead cell that needs to be removed and eat it and digest it to get rid of it. Nerve cells have their own completely different structure. They take in signals from here, decide whether it's important enough to send it on down the line. They send it out here and turn on a bunch of other cells. A sperm cell carries the DNA from the father in here. It has a flagellum on the end that flips back and forth and allows the sperm to swim upstream. And in elementary or middle school biology, we looked at cells under the microscope, and it looked like a little saran wrap bubble with a dot in the middle that was the nucleus. But a cell is a single-celled animal. And look how packed it is with stuff. It's not empty at all, but in fact it's full of things. It has the cell membrane, which is designed to keep out the bad stuff and let in the good stuff. Then it has all kinds of organelles that are designed to do the job inside the cell that needs to be done. First of all, to keep it alive, and secondly, to allow it to do the job it has to do for us. So a muscle cell will have a lot of contractile proteins that pull together and allow it to shorten. A secretory cell that secrete thyroid hormone will have these Golgi Apparatus bubbles that will secrete stuff and then pop and release the hormone into the bloodstream. First of all, especially for anatomy and physiology, the most important part of the cell is the cell membrane. The cell membrane isn't just saran wrap. It's an extremely complicated layer that keeps out the bad stuff, lets in the good stuff, and then controls movement back and forth. And it has receptors on the surface that can receive a hormone signal and cause the cell to ramp up its job, doing whatever it does. You can see it looks like these are little beach balls with tails hanging down in the middle. These are called phospholipids, and they create a phospholipid bilayer. This phospho head is water-loving, but the lipid tails are fat-loving, and they prevent water from moving through. So you got water on the outside because your fluids, your body fluids are water. You have water on the inside because the inside of the cell is filled with water-soluble stuff. But the lipid bilayer here prevents this stuff from getting into here unless you open a little channel and let it go. So this phospholipid bilayer allows only the things to pass that you want to pass. First of all, water passes real easy, not because water can go through the fat, but because there are little water channels everywhere. So water gets to go wherever it wants. Gases cross easily because those molecules are so small nothing can stop them. They're just so absolutely tiny that nothing can stop them. And then fats can cross because it's easy for them to get between these guys, and they can pass right through here because fat loves fat. So a fat-soluble substance will just cross whenever it wants to. Most other things, though, can't. And since your blood is liquid, it's water filled with stuff, the stuff in your blood is water-soluble, and therefore almost everything out here can't get through unless the cell membrane decides it should come through. Let's look at these organelles. Again, these are the organs of the cell. First of all, they don't just bang around and float around in there. They're held together by what's called the cytoskeleton. These are fibers that attach all of the stuff in place so that it stays where it's supposed to. And you have these microtubules that allow you to send a signaling molecule from here all the way over to here if you need it to go. So the cytoskeleton and microtubules form this matrix out here, and the rest of this is primarily water out here. The first of the organelles are your mitochondria. And if you know what a paramecium looks like, it's a single-celled animal. The mitochondria looks like a single-celled animal. And in fact, it is, or was. There was a time when mitochondria didn't live inside anybody else. They were single-celled animals. But forever ago, almost every living creature allowed mitochondria to come in and live inside them because mitochondria have this great ability to produce more energy than they use. And so these are our little energy factories. They take all of our food that we eat, they take what they need, and they give off extra energy. And that's where all of our aerobic energy comes from, our mitochondria. And they are, they can't live outside the body anymore. They have long adapted to just be part of almost all living creatures. But they're really cool. They even have their own DNA. They self-replicate like a single-celled animal would. And so they have their own DNA. They self-replicate. And it's actually not unusual at all for us to have critters that are helping us anyway. Our microbiome in our GI tract is so full of helpful bacteria that if all the bacteria in your body got to vote, and then every cell in your body got to vote, the bacteria would win 10-1. There's that many. And so, for us to live, we need help. And we get it from these guys. Some other organelles, ribosomes, are part of protein synthesis. When you make proteins inside the nucleus, you start the process inside the nucleus. The first half of protein synthesis happens in the nucleus. Then those substances, the RNA, moves out and associates with a ribosome that brings together some more RNAs and forms a copy of that becomes a protein, a basic protein for you. So ribosomes are protein synthesis. The endoplasmic reticulum is this big bunch of flat tubes throughout your cell, and it carries off a lot of different jobs. It transports things and whatever, but you have two kinds. One is called the rough endoplasmic reticulum, or the rough ER, and that's because it has ribosomes all over it. It looks like it's covered with BBs. So the rough endoplasmic reticulum is primarily part of your protein synthesis possibility. You have a smooth endoplasmic reticulum that doesn't have any ribosomes on it, and that helps you create and package some things. It also helps you digest some fats. There are various things that the smooth ER can do. One thing that it really does for us in A&P is it stores calcium for you. Calcium, calcium, calcium. So, for instance, in a muscle cell, to get the muscle to contract, you have to release calcium to turn things on. Well, that's where it comes from, is the smooth endoplasmic reticulum at that point. The Golgi apparatus is right outside the endoplasmic reticulum, and so what you get are the things that are produced in the nucleus, and they are the process finishes in the rough endoplasmic reticulum with all the ribosomes on it. It dumps it into the Golgi apparatus, which is your packaging and FedEx system. The Golgi apparatus will package these proteins and semi-proteins into their final form. It will form these little bubbles full of stuff, and it'll take it to somewhere else in the cell, or it will take it to the surface of the cell and pop and let it go into your bloodstream. So the Golgi apparatus is your packaging and distribution center. Here we go. Start the process in the nucleus. Finish the process on the ribosomes in the rough endoplasmic reticulum. Move those products into the Golgi apparatus, which finishes their processing, puts them into bubbles, and then takes them where they need to go. This one's going inside the cell. This one's going outside the cell to secrete whatever that product was that you just created. Lysosomes are your digestive system. They have enzymes inside that allow them to take in food particles from the outside, digest them for you, and then release the products that the cell needs to do its job. Digestion. And here, every once in a while I do one of these and people love them. Look, mitochondria use oxygen to produce energy. Ribosomes become part of protein synthesis. The rough ER is covered with ribosomes. Smooth ER is not. Stores calcium, and so forth. So make sure that you know the bottom line about each one of these organelles. Because if somebody has a problem with their lysosomes, then what's going to happen to the cell? It can't digest food. It can't do its job. What if you're low on mitochondria? You can't make energy. Okay, so we have the cell floating in this fluid that we produce. It's full of food, it's full of oxygen, it's full of water, it's full of other things that we might need. It's hormones and signaling molecules and stuff. So some things can bind to the outside of the cell and signal the cell to turn on, but other things need to go inside the cell. And so, most of the things out there are water soluble because they're dissolved in the blood. They can't cross the membrane unless the membrane has some sort of a channel or gate or something to allow them to come in. So you have several different mechanisms that we can use to get substance to cross our cell membranes and enter the cell. First of all, permeability means how easy it is for stuff to move. A cell membrane that's really permeable to iron allows a lot of iron to go in. If it's impermeable to something, that means it can't cross. Now, generally, it's permeable to lipids. Lipids are, fats, and therefore fats like fat, and so that phospholipid bilayer doesn't stop them, so they come across. They're impermeable to things that love water. Hydrophilic means they are water loving. So they don't move, they don't allow the water soluble or water loving substances to cross, but they do love fat loving stuff to cross. Impermeable to water, but because of the fat layer, except there are so many water channels that water can move whenever it wants to. So we'll see later how we move water when we need to move it somewhere. Then the membrane contains a bunch of proteins that allow the movement of particles that you want to go, like, for instance, ions, charged particles that will allow the cell to turn on and turn off, sodium, potassium, chloride, calcium. Those are charged particles that are going to activate and inactivate our cells. So we're going to have gates to allow those to come in and out when we want the cell to be activated or inactivated. So, what factors would affect how easy it is for something to cross? Well, if it's fat soluble, it crosses pretty easy. If it isn't, it doesn't. That's a good start. The size and shape, really big molecules don't cross well at all. Little tiny ones like gas molecules do. How thick the membrane is. You might have a cell that really keeps stuff out well by having a super thick membrane. And charged particles like ions don't cross well because the charges in the membrane itself interfere with their ability to pass. So, sodium, potassium, chloride, calcium, bicarbonate, these charged particles don't cross. The membrane itself is impermeable unless it opens up some channels. There's another kind of permeability that we need to talk about, because if you have a layer of cells like that lines your intestine, and you want to get things from the food source into the blood, and you want to control how much comes through, you don't want it squeezing between the cells. You don't want to have big gaps between the cells, otherwise these guys can't control how much food makes it into the blood or at what speed. So the cell membranes themselves are stapled together by what are called tight junctions, these various kind of attachment molecules, occludins, several other ones. Okay? These actually staple the membrane together, so it's really hard for things to squeeze through here. Therefore, the cell itself gets to allow what comes in and what goes out, so it can control the speed of transport. Tight junctions, no tight junction is 100% tight. A little bit of stuff can squeeze through. But it depends on where you are in the body. Some are pretty darn tight. The blood-brain barrier is really tight. The kidneys is pretty loose. It allows a decent amount of stuff to go through on its own. Okay? But still, a tight junction means that the membranes are attached together rather than just sitting side by side. The transport mechanisms we have to get things across the cell then are simple diffusion. It means if you've got more over here than you have over here, it moves that way. Okay, if you got a lot of sugar outside and you got no sugar inside, it's going to go across the membrane some, the best it can, because you always are trying to move down the concentration gradient, so it will just go through the best it can. It's driven by the concentration gradient of that substance. Lipids go that way. If you got a lot of fat over here, not much here, it goes that way. Gases, if you got a lot of oxygen here, not much over here, it moves that way. Facilitated diffusion happens if you have molecules that can't really get through the membrane very well because they're either too big or they have a charge, and therefore you open a hole and let it go through when you want to. It's still driven by the concentration. You know, if you got more something here than you have here, if you open a hole, it'll go through and it will diffuse in the direction down the concentration gradient. So that's facilitated diffusion. You do ions, do that a lot. The ions want to move, and as soon as you open a hole, they just flow on through. Don't do that. Active transport means that you're pushing something that doesn't want to go. I already have a lot of sugar in my cell, but I want even more. And therefore, I will use energy to run a transporter that will push that molecule against maybe a concentration gradient. Maybe there's already more inside the cell than outside, but you want even more to go in. You're going to have to push it against its concentration gradient now. And so you're going to use active transport. Secondary active transport, it's a different mechanism for doing it, but it accomplishes the same thing. You're taking something that wouldn't otherwise go. If you just open a hole, it wouldn't go. So you're using some sort of power to move that molecule from over here to over here against its free will. There are two kinds of secondary active transport. Co-transport or symport means that you're using one molecule going this way to pull another molecule that's going the same way across. Symport, co-transport. I'm going here, I'm taking you with me. Countertransport or antiport is when you have something that wants to go this way, and what it's going to do is power a little merry-go-round because it wants this guy out. So when this guy goes in, he provides the power of this guy to go out. So they're going in opposite directions, so that's countertransport. Co-transport is the same direction. Countertransport is going in opposite directions. Let's look at what these look like a little bit. Simple diffusion. You got carbon dioxide. If you got more here, it's going to go that way. If you have more water here, it's going to go that way. You got oxygen, it's going to go that way. Okay, it's going down its gradient. It's simply going down the gradient because it can cross the membrane. Facilitated diffusion is for things that still will go down that gradient. They'll go down the gradient from high pressure to low, from high concentration to low concentration, from being pushed by electricity. You know, positive charges repel and opposite charges attract. So that might be a charged particle that's being pulled this way by electrical charges. But it can't cross unless you open a hole. Facilitated diffusion, you open a hole and let it go through. Now, something called saturation is possible here because if you have a number of transporters, once they're all open, you can't move it any faster. Even if you've got a bunch of stuff over here, if you only have 8 holes, you know, it's like 8 doorways with a huge crowd over here. They can only go through so fast. So you can saturate and max out your ability to move stuff with facilitated diffusion. Here's facilitated diffusion. Here we have extracellular, intracellular. I want glucose inside. Glucose come here, it sticks here. The thing goes, and the glucose pops out the other side. And then it closes again. It recocks itself, and it closes in the other direction again, ready for another glucose molecule to show up. As soon as it does, it'll open and let it go through. Okay, primary active transport happens when you expend energy at the transporter to push molecules in the direction they don't want to go. Okay, adenosine triphosphate. We'll talk about this a little bit more later, but adenosine triphosphate is our energy molecule. It's an adenosine with 3 phosphates stuck onto it, and that last phosphate has a high energy bond, and when you break that bond, you get a poof of energy that gives you enough power to do something, like move a molecule in a direction it doesn't want to go, to cause a muscle to contract. So we use ATP energy to run these active transporters. These are so important that 80% of your resting metabolic rate is just these transporters using up energy just to keep stuff where it's supposed to be in your body, on this side of the membrane instead of that side. If you got too much over here, you move it over this way. You got too much over here, you move it that way. And keeping stuff on the right side of the membrane uses up most of our resting energy. Our best example is, and we're going to see this forever, so you got to know this one, the sodium-potassium ATPase pump. This means it uses the enzyme that breaks apart ATP to give you energy. So you're using ATP here to move sodium one way and potassium the other. Our extracellular fluid has a lot of sodium in it and hardly any inside the cell. Potassium is inside the cell, hardly any is outside. And the whole reason those are where they are is because these pumps work 24/7 like the Energizer Bunny, pumping out sodium that leaks in and pumping in potassium that leaks out. So it keeps a high concentration of sodium outside, high concentration of potassium inside. These are critical for maintaining our gradients of these ions that are going to turn muscles on and off. Sodium potassium pump. ATP, it tells you it uses ATP. You also have a calcium ATP pump that pumps calcium back into that smooth endoplasmic reticulum after it's been released during muscle contraction. Here we have primary active transport. Here we have sodium being pumped this way. We have potassium being pumped this way. Potassium leaks back in, has to be pumped out. Sodium leaks out, has to be pumped back in. You're keeping sodium here, you're keeping potassium there. And you're using ATP. You're breaking off one of those P's. You're using ATP right here. You're breaking off one of those P's and getting your energy that causes these molecules to move. Now remember, the secondary active transporters also are pushing stuff in directions they don't want to go. Co-transport moves in the same direction. Counter-transport moves in the opposite. What they usually use is the power of sodium. Now look back here. We got all this sodium here. If I'm keeping the sodium here on purpose, I want to have a lot of sodium here because I might need to move something this way that doesn't want to go. And I can use this high pressure amount of sodium to combine with, say, glucose and move it across the membrane if it doesn't already want to go that way. So you're usually using the gradient of sodium, which is real powerful, to run a transporter to move some other molecule that doesn't want to move. Here we have, you want to get more glucose in here. You already have a bunch, so glucose wouldn't go if you just opened a hole. But if glucose binds and sodium binds, the power of sodium will cause this change of shape and the sodium comes in, and the glucose comes in. Now, pretty soon you'd have too much sodium in here, right? But guess what? We have sodium-potassium pumps. It keeps pumping the sodium out so you can do it again. Pump it out, you can use it again. Pump it out so you can use it again. This is co-transport. They're both going in the same direction. Counter-transport, remember, we go in the opposite direction. What you would be doing is binding sodium here. You'd be binding something like calcium. You want to get calcium out. And then the thing rotates around and the calcium pops out here and the sodium pops in there. So that would be counter-transport. There's a lot more co-transporters in your body, though. You're always doing this with sodium. As a result of all of these actions, and especially the action of the active transporters like the ATPase pump and the calcium pump, you maintain these concentration gradients that are going to become really important to us when we start talking about turning cells on and off. We're going to cause electrical ion flows that will cause our cells to turn on and off. So our extracellular fluid, that's everything outside the cell, is mostly sodium. All that sodium keeps getting pumped out. Tons of sodium. Very little potassium because it's all being pumped in here. Look how much potassium. Potassium is in here. Sodium is all being pushed this way, so it's all out here. Potassium is being pushed this way, so it's all in here. Calcium is being pushed out too. It's all outside. There's not as much of it. Sodium's the big player because look how much we have. There's a ton of sodium out there. So we can use it to run our transporters and stuff with because that's a huge concentration gradient that's trying to move the sodium into the cell. So you can use it to move glucose in with a co-transporter, for instance. You don't need to memorize these numbers, but do memorize that sodium is way more outside than inside, potassium is way more inside than outside, and calcium is way more outside than inside. That's critical to understanding how we're going to turn and turn off the cells. And if the cell membrane needs something to move across that won't fit through any transporter because it's too big, you can engage in phagocytosis, which is cell eating. This is like those amoeba type, white blood cells. They just surround a bacterium. A bacterium is way too big to cross through a transporter, so the cell just eats it and brings it in. Pinocytosis, the cell membrane just makes a little hole here. Stuff goes in it, and then it pops and forms a little bubble inside that has that stuff in it. So pinocytosis is cell drinking. Phagocytosis, big stuff comes in. Pinocytosis, smaller, but still too big to go through a gate. Endocytosis, receptor-mediated, means when whatever this thing is that the cell wants actually bumps into the cell, it turns on a receptor that causes the cell to wrap around it and eat it. Exocytosis is the process of the cell getting rid of big stuff. Like we saw with the Golgi apparatus when it made a bunch of hormones and then it just goes to the cell membrane and pops and explodes outward and all the stuff goes outside. That's exocytosis. So endocytosis includes phagocytosis and pinocytosis. Those are all coming in. Exocytosis is going out. Here we have endocytosis. Something bumps into these receptors here, and they start bending, and it bends even more, and then it closes off up here, and the bubble of that substance winds up inside the cell. Here's exocytosis. Okay? Here we had, oh no, this is pinocytosis. We're going to show exocytosis in another slide or even another lecture. But here we have endocytosis, very similar to that artist drawing here. here these substances are binding to the receptors, and it bends in, and it closes off, and pop. There you have your substance inside the cell. So this is the same one we saw before, but this is real life. And transcytosis means it's got to come in one membrane and go out the other one. This is like that intestinal wall. It's got to come in one membrane and go out the other. It's not just coming in the cell and staying there. This cell has to suck it in and spit it out. It's got an innie and an outie on it, so that you'll take it in one way and exit from the other side. So here we have, we had our sodium coming in and being pumped this way. Now we're pulling sodium hard enough that we can use this sodium to run a co-transporter to pull glucose in. And now that you've got a lot of glucose in, you can just open a hole here and let glucose go out. So you have an innie for the glucose here, and you have an outie here, and it's taking glucose from your intestine to the blood. And here you're using a couple of different mechanisms to get it in one side and out the other. Okay, catch your breath a second, because that is how we move stuff. Solute. Solute is anything that's dissolved in the blood or in the plasma or in the interstitial fluid. If you dissolve something in water, that's the solute. The water then is water. It has to move also. And water, because we have so many holes that it can go back and forth whenever it wants, if we want to move it, we can't pump it because it'll just go back through a hole. So if we want water to move from one side of a membrane to another, we're going to have a mechanism for moving it across. And this is going to be osmotic pressure. First of all, you have to understand what molarity is. A mole of a substance is its gram molecular weight. It's however much 6.02 times 10 to the 23rd molecules of that stuff happens to weigh. Because hydrogen only has one proton, a mole of hydrogen weighs a gram. Oxygen has 18 protons and neutrons, so a gram of oxygen weighs 18 grams. A mole of oxygen. So the way we do this, like if you have a mole of carbon dioxide, you'd have 12 grams of carbon because it has 12 protons and neutrons. I said 18, I mean 16. 16 for oxygen. 16 times 2 is 32, plus 12 is 44. So a mole of carbon dioxide would be heavier. It's got 2 molecules in it. It weighs 44 grams per mole. Okay, so that's just a quick review on what a mole is. It's just a lot of molecules, but molecules are so light that you can actually weigh a mole. It's like a dozen eggs. A mole of molecules is a certain number, and you know how much they weigh by how big they are. Molarity, then, is how many moles of a substance are in a liter of water. If I have 1 mole of sugar in a liter of water, it's considered to be a 1 molar solution. If I have a 1 millimole, a thousandth of a mole, it's a 1 millimolar solution. Millimolar, you might have, 100 millimoles of a substance you put in, and then you have 100 millimolar solution. Okay, so it's just how much of that stuff happens to be in every liter of your liquid. Now, osmolarity, first of all, let's talk about osmosis. Osmosis, let's start again. If I have a area of fluid here, and I have an area of fluid here. See if I can get here. And I have more stuff here than I have here. Stuff is going to want to move that way because more stuff goes that way. And that's diffusion. So you would have the same concentration there everywhere. All right? But what if you have a membrane in the middle that doesn't let the stuff move? Your body or nature or chemistry likes everything to be absolutely even. It likes the same osmolarity. It likes the same pressure on each side. So if I have a lot of sugar over here, and I have not very much sugar here, and I have a membrane in between that won't let the sugar move, water will move to try to dilute this thick one and concentrate the thin one so they're even. So osmosis is a force that draws water toward any high concentration of stuff. If my blood has too much stuff in it, it will osmotically pull water out of your cells to replenish your blood, and your cells will shrink and get dehydrated. It happens if you sweat too much. Your blood gets thick, and it starts pulling water out of cells by osmosis to try to keep your blood thin enough that your heart can beat it. So osmolarity is the molarity of a fluid and therefore its effect on the osmotic forces. So generally speaking, if I have a 1 millimolar solution, it's a 1 milliosmolar solution. The only changes are if it splits and has multiple, effects after it gets in the blood, but we won't worry about that right now. We're talking about, I have this amount of osmolarity here, and I have this amount here, and wherever it's highest, it will pull water toward it. And that's how we move water. If I want water to go that way, what I'll do is I'll pump some stuff over there, create an osmotic gradient that will suck water across. Osmosis sucks. Most students agree. Osmosis sucks. Osmosis pulls water toward it. Now, knowing that, your blood and all of your body fluids are about 300 milliosmoles per liter of water. That's their concentration of stuff. About 1/3 of a mole of all of the things together, sodium and potassium and chloride and everything else that happens to be in your plasma, adds up to about 300 milliosmoles per liter. Your interstitial fluid, which is outside the blood vessels, is also about 300. If this were 350, it would pull water from the blood into the cells. If the blood were 350, it would pull water from the cells into the blood, because remember, osmosis sucks. So the higher osmolarity would draw water toward it. But remember that your blood is supposed to be, and all of your body fluids are supposed to be, about 300 milliosmoles per liter. That's the concentration of stuff in our blood normally. So, you have a membrane that's permeable to water but not the solute. So you need to move the water to achieve a concentration balance. Osmosis sucks. The water moves toward the high concentration to dilute it so that they become as even as they can. Osmotic pressure is how hard it's trying to pull. It's based on the total amount of solute. It doesn't matter that part of it is a big molecule and part is a little molecule. Part of it is hydrogen and part of it is glucose. It's the total number. It's only number that matters. So 300 is bigger than 250. 300 will pull water toward it. Water flows toward the osmotic gradient. It flows down water pressure. If you put too much pressure in there, it goes out. But if you have a lot of osmotic pressure, it pulls it in. one osmolar solution actually exerts 373 pounds per square inch. It's really a powerful force. And so we have about a third of that. That's still 100 pounds per square inch of osmotic force in our blood. Fortunately for us, it's the same on the outside of the blood vessel too, so they're even. But if you had a blood vessel that only had it had 0 and outside had 300, the blood vessel would just probably blow apart from the force of trying to pull the water out so fast. So it's not an insignificant force, and it's the way that we can move water around in our body. Move stuff over here, the water will be pulled by osmosis. Fluids that are isotonic means they have the same osmotic force. So if you put isotonic saline in somebody's blood vessels, it'll just add blood volume, but it won't suck water out of their cells, or it won't blow water into them. It would be, isotonic means 300, 300. That's good. A hypertonic solution means it's more highly osmotic. So if I put 350 osmotic solution in my vein, it would be hypertonic to the cells, and it would pull water out of the cells. Probably not what you want to happen. That's why we use isotonic saline. A hypotonic solution means it's too low. So if I put 250, if I put plasma in that had only 250 milliosmoles, then the cells would be hypertonic, and they would suck too much of that fluid into them, and they would explode. So you want a isotonic solution going into your blood vessels. Should be about 300 inside and out, mostly sodium and chloride on the outside, mostly potassium on the inside, causing the osmotic pressure to be about the same inside and out. If you do put a cell in a hypertonic solution, that hypertonic solution will suck water out of the cell and it'll shrink. Sweat. If you sweat, you lose some electrolytes, but you lose mostly water. So if you sweat too much, the blood gets too thick, and it starts pulling water out of the cells. The cells shrink, and they start to malfunction. You get muscle cramps, you get headaches. Here we have a normal red blood cell. We've dropped it in some water that is hypertonic, and it sucked water out of the cell and caused it to collapse. But if you put it in a hypotonic solution, the tonicity out here is too low, osmolarity in here is too high, and it sucks water in, and it swells up and pops. If I can demonstrate that in an online lab, I'll try, but we do it when we have the live lab. I actually take some of my blood and I drop some in distilled water, and it does this, because there's no osmolarity out here, so the cell keeps sucking water into the cell where the osmolarity is higher, 300, right? Here I put it in salt water. Now the osmolarity out here is too high, so it pulls the water out and it collapses. And therefore, as I said, water follows solute. Since we mostly have sodium available, tons of it, if I want to move some water across a membrane, I'll use a sodium pump of some kind. I'll move sodium over, and the water will be pulled across by osmosis. Here we go. I'm pumping sodium this way. The water is being pulled by osmosis to follow the sodium. I've used sodium pumps to move the sodium this way, and therefore the water is pulled across by osmosis. It's the way we absorb a lot of our water in our intestine. We move sodium across, and water follows. So we don't have diarrhea. Instead, we absorb the water. Cystic fibrosis is a fantastic example of osmosis gone wrong. In the lining of our airways, we make mucus all the time because we need bad stuff to stick to the mucus so it doesn't go all the way in our lungs. And then we have nice little cilia that are constantly doing this and moving the mucus out, and the mucus moves out and carries all the crap out with it, and you spit it out or you swallow it and your stomach acid kills it or whatever. But this mucus is really thick stuff. It's really thick. So what our body does is we have chloride pumps, and they pump chloride across. Sodium and chloride love each other. Negative, positive, they attract. So when the negative charge goes, sodium finds its way across too, and now you have a lot of sodium and chloride going across. Water gets pulled by osmosis, and it keeps the goo soft enough that it can move out. Cystic fibrosis, this chloride channel is actually called the cystic fibrosis transporter. In a person with cystic fibrosis, a genetic abnormality means this chloride channel doesn't work. They either don't have them or it doesn't work. So if you stop pumping chloride, no sodium goes, no water gets pulled across, and the mucus gets so thick it won't move. And the person drowns in their own goo. And so you beat on their back, or you put a vest on them that vibrates and knocks it loose, and they try to cough it out, and it's a really unfortunate, really difficult condition to deal with. They're getting better at it. They have some additives and things that they can do. But here we have the movement of ions causing the movement of water that is keeping things nice and soft and moist. And here, the lack of movement of ions cause a lack of move of water and cystic fibrosis, thick mucus. Various things that result from problems with osmosis. Kidneys. Kidneys transport solutes and water into the urine so you can pee it out. And what they are supposed to do is have you pee the right amount of solute to get rid of things you don't want, pee out the right amount of water to keep your blood volume right, and everything is all fine. But if you pee out too much water, you will become dehydrated. If you pee out too much solute, you'll become hypohydrated. And in some cases, if the solute goes out that's not supposed to, it pulls water by osmosis, causing you to pee all the time. Okay? Diabetes works that way. Sugar. You have so much sugar in your blood, it winds up in your pee. You pee out sugar, it drags water by osmosis. You pee a huge amount and you're dehydrated and thirsty all the time. It's just being pulled by osmosis. A sprained ankle. When you sprain your ankle swells up. It doesn't fill up with blood, it fills up with water. Because a lot of plasma proteins and things that are supposed to stay inside the blood vessel to osmotically keep blood in, now all of those proteins and things are released out into the tissue, and their osmotic forces pull additional water out of all the surrounding blood vessels, and your ankle swells up it's like a water balloon. And it's because these plasma proteins cause too much osmosis out there, and they pulled water into your ankle, and your ankle swell up. Pulmonary edema we're going to talk about later. Osmotic diarrhea, though, if you have if you don't digest food properly. And so instead of absorbing the food and having the water pulled by osmosis, the food stays in your intestine. It pulls more water into the intestine and you have diarrhea. Okay? You want your food to get absorbed so that it will help your water get absorbed, not the other way around. And that's it. Again, that's a lot of stuff. The cell membrane, how it works, the organelles, the transport mechanisms, simple diffusion, facilitated diffusion, and then your active transporters using ATP energy in some fashion. Now remember, a primary active transporter actually uses ATP right there. A secondary transporter uses the concentration of gradient of sodium to run the transporter, so there's no ATP there. But you had to use ATP to get the sodium there in the first place. That's why it's secondary. So the ATP pumps are maintaining the sodium gradient. The sodium gradient runs the secondary transporter, thus secondary active transport. Co-transporters, symporters, and antiporters or exchangers. And then osmosis. Move the solute. Water follows by osmosis. Hypertonic, too much osmolarity will pull water toward it. Hypotonic, too low, will pull water that way. Too low, it will be removed from that area. Sorry, I didn't say that well. A hypotonic solution will allow water to be pulled into a hypertonic area. Hypotonic plasma would cause fluid to be pulled into your cells. Hypertonic plasma would pull water out of the cells into the blood. It's wherever it's highest. Wherever it's highest. Okay, best of luck.